Back in another video tutorial from microbiology with Charlie. In that video tutorial we are going to discuss about our affinity chromatography and ion exchange chromatography. So here, before going to talk about affinity chromatography and ion exchange chromatography, I want to revise our previous class where we have discussed about paper chromatography and thin layer chromatography, the two simplest type of chromatographic technique. So pen paper chromatography, I again want to mention you that we are using a solid phase, where we are considering solid phase a chromatographic strip, right? So this in that chromatographic strip we are placing a small dot or line, and this small dot is nothing but our sample in a mixture of the solution, right? And then we are placing our strip into a jar which contain a shallow layer of solvent, and this solvent via capillary action help us to separate our molecule, right? So here the separation is entirely on the basis of their size and shape. Okay, so paper chromatography is the most easiest and simplest method. That is that is what it advantage, and it is also cheap method. Okay, and it allow you to identify unknown inorganic and organic material with the help of the RF value we have discussed, that is the retardation factor. That is what? That is the distance traveled by the substance and distance traveled by the solvent font. So suppose this green line is the solvent, so I am denoting it as B, and the yellow line here is the our substance, right? So sorry for that, I made a mistake. That suppose this one is our substance, and sorry, this one is the solvent and this one is the substance. That what I want to told you, that this is your mobile phase. This mobile phase will undergo capillary action, and this capillary action will cause the rising of the mobile phase through the paper. Okay, and during this rising it will also force the sample to rise up the paper. Okay, so now it is obvious that the mobile phase will move larger or longer than our substance. So that means that this green line or green dot that we are seeing is our substance that we are want to separating, and this one is the solvent front. So our RF value will be, if we are denoting this as A and this one as B, then our RF value will be RF is equal to A by B. And with the help of such kind of RF value we can recognize unknown inorganic and organic material. Okay, so that is the major advantage of both paper chromatography and thin layer chromatography. As you can see that here they are also providing us to calculate the RF value, right? So now let's move to the another kind of advantages for paper chromatography. So paper chromatography also allow us to use a very less quantitative material for separation. Okay, that cannot be possible in more advanced type of chromatography. Okay, and that's all for advantage part of the paper chromatography. And disadvantages, what are the disadvantages of the paper chromatography? So paper chromatography you cannot use for quantitative analysis of larger sample, right? That is as well as the disadvantages of thin layer chromatography too, because you can see here the paper length is limited, right? So separation will be done up to a certain length. After that it will top, right? So separation cannot be done at a higher extent. That is why you cannot separate with a very large sample with the help of paper chromatography or thin layer chromatography, but you may prefer separation of small things such as amino acid mixtures. You can easily separate with the help of paper chromatography, thin layer chromatography. So now, as I am telling repeatedly that paper chromatography and thin layer chromatography are same, then why we are studying it separately, right? So that simply means that there will be some difference between the paper chromatography and thin layer chromatography, right? So let's discuss about it. So the thin layer chromatography is the slight modification of the paper chromatography, because in paper chromatography there is a problem. The problem is paper chromatography takes a significant amount of time for separation, okay, and here we have to confine on the chromatographic paper as a solid phase. We cannot change chromatographic paper with any other thing in case of paper chromatography. So thin layer chromatography solves those kind of problems. So thin layer chromatography is nothing but here we are using a thin layer absorbent that can be made up of cellulose, alumina or silica as a solid phase. Okay, so and the rest of the process is same, but the presence of thin layer adsorbent as a solid phase provide us the opportunity to use different kind of solid phase, right? Not only that, it also provide us a better separation, okay, that cannot be done with the help of paper chromatography, and it also provide us a faster separation, okay, that also paper chromatography cannot provide. But still it cannot separate a very large sample, and also importantly here the separation is going on in open air, so there is a high chance of contamination, okay. And here another thing I want to mention you, that here the solvent that we are using is a mixture of acetic acid, butanol and H2O in a ratio of 8:2:1. That is a very important for MCQ purpose, okay. They can tell you about the general composition of the solvent for thin layer chromatography. So thin layer chromatography is especially used for amino acid separation. A process called two-dimensional chromatography along with separation of vitamin can also be done, okay. So after separation we can visualize our protein sample by using visualizing reagent, okay. So now after completing this part, let's move about our next topic, that is the column chromatography, okay. So column chromatography solves a major disadvantage of paper chromatography and our thin layer chromatography, that is they cannot provide larger sample separation, right? So instead of using paper or silica cellulose adsorbent papers, now we have moved towards column, okay. So let's understand about the column chromatography principle. So column chromatography is again a based on the interaction of the stationary phase and mobile phase. So here the stationary phase is a porous solid matrix which contain some specialized chemical property, okay, on the basis of which we are separating our samples on the basis of different things, right? So column chromatography can separate protein on the basis of their size, on the basis of their charge, on the basis of their binding affinity and many things, okay. But the idea is the fact on the basis of which you are want to separating your protein, that fact should be utilizable by the stationary phase for your separation, okay. That is the main concept of column chromatography, that you are using a solid phase which is nothing but a porous solid matrix, and you have layered your mixture on the top of the column, then your column will percolate through the solid phase, okay, as a ever expanding band from the larger mobile phase, and separates on the basis of their particular property you are using for separation, okay. Then by using visualizing here, another important thing is I want to mention you that here the first thing we want to do is the removal of the unneeded material from the mixture, okay. After that there will be only our sample. Suppose this one is our sample, then we will use a elution buffer that contain some molecule that have a more good growth property, or that have a more good affinity to the solid matrix than the protein, will be added as a elution buffer. So this elution buffer will help us to isolate our desired protein sample, okay. So the idea is we are constructing a solid phase. That solid phase is nothing but a porous solid matrix. This porous solid matrix contain a appropriate chemical property that we are willing to use to separate our proteins on the basis of that. So then we are loading our sample on the top of the column. The sample will percolate then through the stationary phase and separates depending on the property that we have chosen to separate them, okay. Then we will use a elution buffer. That elution buffer definitely contain something that have a good affinity, obviously more better affinity for the solid matrix than our protein, okay. So this elution buffer will help our protein to be removed after separation, okay. So that is the idea of the column chromatography. So column chromatography, as you can see, it is a best method for purifying protein, okay. Purification in research purpose is often done with the help of this method, and it provides you the choice to use a wide variety of mobile phase. That is exciting thing, but it has some disadvantages too. So what kind of disadvantages? First of all, it is very time taking. Only the packing of the column with the stationary phase takes a larger time, okay, and you have to apply constant attention during the separation. And also the column chromatography is not foolproof as in case of we are paper chromatography, okay. And the most important thing, there is a chance of entrapment of the air bubbles in between the solid phase and the mobile phase as a result of dry packing, which may facilitate crack in the adsorbent layer, okay. So that means air bubble can be formed between the solid phase and mobile phase during your packing step. If dry packing is occurred, then it will ultimately promote cracking in the adsorbent layer that will affect our result. That is why you have to require a good technical skill for packing the column chromatography for separation. Otherwise it can affect your result. That is why it is quite difficult and require good technical skill, okay. So what are the application of the column chromatography? Column chromatography will help you to separate or to purify important component from the non-important components. Then it help you to purify metabolites, important metabolites from a particular mixture, and it will help you to determine phytomenadione in drugs and injections, okay. So that is the concept of the column chromatography. Okay, although we have previously discussed about the column chromatography, right? Now let's move to the next one, that is related to the column chromatography. This one is size exclusion chromatography. Okay, so size exclusion chromatography separate proteins or any kind of biological samples strictly on the basis of their size. Okay, none other than anything. So how the separation is going on? So here we are using a porous polymer beads as a solid matrix. Okay, so I am repeating that here we are using porous polymer bead. Okay, and in case of ionic our column chromatography, we have used porous solid matrix. Okay, this porous solid matrix will help us to separate protein on the basis of the different properties that we have chosen to separate protein, right? But here we are using porous polymer beads. That means here we are using various kind of ball like substances that have pores, and inside the pores our protein can easily penetrate if our protein's size can accommodate the penetration. Okay, so the idea is we will first prefer the solid phase with the help of this porous solid matrix. Okay, after preparation of the solid phase, we will perform our mobile phase, prepare our mobile phase. That mobile phase is nothing but the buffer containing sample, and we will add it on the top of our column. Okay, after that the liquid will percolate through the column, and during this separation the protein molecules whose size is smaller than the average pore size of the polymer beads will enter inside the pores. Okay, and as a result they become entrapped and separated from the liquid phase. Okay, and the components of the mixture whose average pore size is greater than the average pore size of the porous polymer beads can easily removes from the column, or can easily passes through the column without any resistance, right? As a result, I want to mention you a very important thing that you need to keep in your mind, that in size exclusion chromatography you will get larger molecule faster than smaller molecule. That seems quite unusual, right? Because in case of column chromatography we are having smaller molecule faster, after that larger one, right? So here the idea is totally different. That is the importance of that concept, because remember it very carefully, that size exclusion chromatography can only be used when they will told you to separate a mixture where the desired purifying product is small. Okay, in such cases first of all you have to wash out all the larger debris. So you can use size exclusion chromatography, because size exclusion chromatography will entrap smaller particles inside the porous polymer beads for you, and the remaining liquid will be percolates through the solid phase and they will be separated, right? And in case of column chromatography, you will prefer column chromatography when your desired material from the mixture is larger in size. Okay, so column chromatography will help you to first remove the smaller particles, then you will end up with only the larger particles, and then by using elution buffer you will have your larger particle, right? So that is the idea of size exclusion chromatography and why you have to use size exclusion chromatography. Although the size exclusion chromatography's resolution is not very high, and that is why it is often employed as the last polishing step of purification. Okay, so it help you to determine the tertiary and quaternary structure of the protein when we are performing the entire experiment under native condition without any denaturing agent that can promote protein denaturation. It will help you to separate oligomers. Okay, it will help you to separate the homologs such as sugar from juices. Okay, and it will also help you to separate determination of the molecular weight. Okay, and another important thing that I want to mention as an advantage of the size exclusion chromatography is size exclusion chromatography provide you a faster separation that column chromatography cannot, a relatively faster separation. Okay, and here the separation is done with a more or less good sensitivity. Okay, although it is not too much high. So now let's talk about the disadvantages of the column chromatography. One disadvantage I have already mentioned, that it has low resolution and there will be limited number of peaks, and at least 10% differences in molecular mass can be separated with the help of this size exclusion chromatography. Molecular difference of less than 10% cannot be separated by size exclusion chromatography. On the other hand, in column exchange chromatography, if you are taking another kind of properties as your protocol to separate those materials, you can easily separate, but size exclusion chromatography under such cases cannot help you, right? So that is the idea of the size exclusion and column chromatography. So if in your examination they will ask you about write a difference between the size exclusion chromatography and column exchange chromatography, so the first thing that you have to mention that column exchange chromatography separate protein on the basis of different properties, right? It can be size, it can be charge, it can be binding affinity, it can be anything. On the other hand, size exclusion chromatography specifically separate protein on the basis of their size. Okay, another thing that I want to mention you, that column chromatography provide larger molecule fast than the column chromatography will provide smaller molecule first than the larger molecule. Larger molecule will be entrapped in the solid phase. But in case of your size exclusion chromatography, smaller molecule will be removed faster and larger molecule will be entrapped. That is the very important thing. Okay, and it is your column chromatography is a preferable method for purification, but size exclusion is not because of its low resolution, right? And column chromatography will takes time for separation. On the other hand, size exclusion chromatography takes lesser time for separation than the column chromatography. Okay, so that's how you can create a difference between column chromatography and size exclusion chromatography. Now today's topic, that is our affinity chromatography. Okay, so let me discuss. So today first we are going to discuss about ion exchange chromatography, then affinity chromatography. Okay, so ion exchange chromatography. In ion exchange chromatography, we are using differences in charge, that is whether your protein is positive or negatively charged and what is the magnitude of the charge. Okay, so here we are simply separating protein on the basis of its charge, and I hope you know that protein's charge is determined by the pH of its environment, right? So Ion exchange chromatography separates proteins specifically on the basis of their charge in the appropriate pH or in the given pH. Okay, that is necessarily you have to mention during your in the examination copy, otherwise it will be uncompleted. That protein's charge is determined by the pH of the environment in which your protein is present. Okay, so here the idea is we are taking a column and here we are using synthetic polymer beads. Okay, so these synthetic polymer beads contain bound charge group. Okay, in such a way. So now the question is, what is the utility of those bound charge groups in the synthetic polymer resin? So this synthetic polymer resin, this bound charge group will help you to separate your protein of interest on the basis of their charge. Let's see how. So first of all, I want to tell you about the basic difference of anion and cation. I have find out that it is a quite tricky one. So Ni means here I am referring as neutral ion. So if this neutral ion gets negative charge, it will be anion. Okay, and if it gets positive charge, it will be cation. Remember this very carefully. So it will help you to remember about the ion exchange chromatography concept. Okay, and I am willing to mention another mnemonic that will help you to remember. Okay, I will mention it later. So for now, just remember that anion is negative and cation is positive. Okay, now let's move. So here we had solid matrix which contain bound charge group. So now here the mobile phase, that means the mixture of the buffer and the sample should have a appropriate pH because we are using buffer, right? So and as we have used the buffer, that means we know what will be the charge of our mixture. Okay, suppose we have lysine and suppose we have a protein which is lysine rich, and along with we have a protein which is aspartic acid rich. Okay, and here I want to purify lysine rich protein. Okay, so we all know that lysine is a basic amino acid, isn't it? So here I want to touch an important concept, that is the pi value of the protein. Okay, so pi value is the pH in which protein do not migrate towards any of the electrode. Okay, so if your environmental pH is greater than the pi of your protein, that means what? That means your environment become basic, right? And under such circumstance, there will be too much OH minus ion in the environment. Okay, and in that case, the OH minus ion will be converted into H2O by the H of the COOH. I hope you know what is the basic structure of amino acid stuff, that it contains a carboxylic acid group, right? So as a result, the protein become negatively charged, right? Isn't it? Yes. So we are willing to separate lysine, right? So our goal will be to trap lysine and allow aspartic acid to pass through the column. Okay, so our goal will be to trap the lysine, right? So how we can trap with this? So the bound charge particle we are using should have a negative charge, right? Because lysine is a basic amino acid, that means it predominates positive charge. Okay, so we have to use a negative charge containing bound molecules. Okay, so by the help of that we are using negatively charged bound molecules in the synthetic resin, and we make the positively charged lysine with the help of buffer that we are using. Okay, I hope up to that point it is very clear to you. Then we will add our protein mixture. So aspartic acid will exhibit near about no hindrance and it will pass through the column, but here lysine will face the hindrance and it will ultimately become trapped in the solid phase. Okay, that is how you have separated lysine from aspartic acid. But now the question is, how can you isolate your lysine? Whether you will going to break the column to isolate your lysine, is it? No. After the separation we will use a elution buffer that obviously contain a more positively charged compound than lysine. So as a result, lysine will lose the competition with those positively charged compound and it will be separated. That's how the entire ion exchange chromatography works. Okay, so as you have seen that depending on the type of the protein we have to use our bound charge groups, right? So there are two kind of ion exchange chromatography that I want to told you, that is, I am mentioning ion exchange chromatography as IC. So one is cation exchange chromatography, I am mentioning it as CC, and another one is anion exchange chromatography. So here I am wanting to mention you a mnemonic that you help to remember that which chromatography contain which bound charge group. So remember, as the alphabet A comes first, then the C, so the anion exchange chromatography will have positively charged group. Okay, and the cation exchange chromatography will have negatively charged group as its solid phase. Okay, so anion exchange chromatography will separate anionic compound for you with the help of those positively charged group, and the cation exchange chromatography will help you to separate positively charged group. An example of anion exchanger is tertiary amine, secondary amine. Okay, where tertiary amine is more stronger than the secondary amine. Okay, and an example of cation exchanger is sulfonic acid, carboxylic acid. Okay, so here I have shown you a example of two charged amino acid. Suppose there will be mixture of different charge groups. Okay, and now you are willing to separate it. So what can you done? So here the idea is you will prepare a gradient of pH. Okay, that will help you to separate. So here I want to mention you another important thing, that which kind of factors influence the binding or the affinity of the protein towards those bound groups. So the first one is the concentration of the free salt ions that are present, because I think I do not have to mention that salt are also charged groups, right? So it can also show interaction with those bound charge groups, right? So the concentration of the free salt ion we have to keep in your mind about them. Okay, and another one is obviously the pH of the environment that will facilitate and determine what charge our protein sample will contain, right? So now let's talk about the factor in affecting ion exchanger. Okay, so the one is the cross linking. If cross-linking is extensive, we have to prevent them, right? Otherwise our ion exchanger will lose its exchange capacity. Okay, and another, so cross linking is down regularly control the exchanging capacity, right? And another one is the swelling capacity. So swelling capacity helps in exchange. Why? Because swelling facilitates the exposure of the charge group, which facilitates the interaction of the charge group with protein. Okay, along with the sample concentration and charge, and buffer charge also play an important role. Okay, so what are the advantage? The first advantage I want to mention you that it is environmental friendly. Okay, where here we are using various kind of binding charge groups, right? And the longer life resin, the resins have longer life that we are using. Synthetic polymer resin, they have long life, and obviously cost effective. Its maintenance is very cheap. But the problem is the nature and properties of the bound charge groups, the ion exchange resin, and the nature of the exchanging ions. These are the disadvantages. So here the advantages are the longer shelf life, longer life of the resin, and cheap maintenance. Okay, and the disadvantages are the nature of the exchanging ions and the nature and properties of the ion exchange resins. Okay, so what are the utility? We can use ion exchange chromatography to separate ionic impurities from the inorganic mixture, from a mixture, right? And we can also use this for the demineralization and deionization of water concentration of the ionic solution. So our ion exchange chromatography's advantages are it have a longer life resin, cheap maintenance, and we are dealing with environmental friendly compounds. And disadvantages are the nature of the exchanging ions and the nature and properties of the ion exchange resin are the disadvantages. Okay, so we are using ion exchange chromatography for the separation of the ionic impurities. We are using them for the concentration of the solution, right? We are using them for softening of water, demineralization and deionization of water. Okay, so that is the concept of ion exchange chromatography. I hope you have understood it very well. Now today's last topic, that is the affinity ligand chromatographic technique. Okay, here we are utilizing a non-covalent interaction between our protein and the ligand metabolite. Okay, so here the interaction is non-covalent. Okay, so now I want to told you that what is ligand. So ligand are nothing but immobilized compound that act as a molecular fish hook. Okay, it have the ability to specifically pick up proteins from a mixture of the sample. Okay, and it act like a substrate in enzyme substrate reaction that can selectively recognize its active site in a enzyme. Okay, that's how ligand can selectively recognize the desired protein. That property we are using. Okay, for example, a interesting type of chromatographic technique, that is NINTA chromatography, that is nickel nitrile triacetic acid chromatographic technique. That is nothing but a affinity chromatographic technique. Okay, so this affinity chromatographic technique, here we are using chelated nickel atom. So how I am going to discuss with you after explaining the basic concept of the affinity chromatography. So here in the solid phase we are using a gel matrix which are nothing but our agarose gel, okay, and which contain the ligand which is specific for our protein that we want to separate from a mixture, okay. So let's take an example. Suppose we are want to separate a enzyme, okay, that is also a protein, and we know what is the substrate specifically in which that particular enzyme acts, okay. So we are using those substrate as a ligand. As a result, when the mobile phase will pass through the solid phase, the substrate ligand will selectively pick up only its desired protein, because it can only bind with the desired protein's active site, and that's how they can provide a extremely sensitive separation with the help of this non-covalent interaction, okay. And again similarly we will saturate our process, or we will complete our process with the help of a elution buffer. This elution buffer will help us to separate our desired material. How? Here we are just putting another enzyme that exhibit a more greater affinity towards that substrate than our protein of interest. As a result, the protein of interest will lose the competition and it will remove, and its place will be taken up by the enzyme. That's how easy our affinity chromatographic technique is. So in case of nickel nitrile or triacetic acid, I have mentioned we are using the same thing. Let's see how. So here we are using a chelated nickel atom, okay, as a chromatographic technique. So here we are can separate protein that have 6 or more histidine residue at its C-terminal end, okay. And I have discussed about that kind of separation various times in our recombinant DNA technology videos, where we are producing GCSF, right? Try to remember about this. I have told you about this in E. coli lac promoter T7 RNA polymerase based vectors. That is the same thing, and it is totally based on the ion exchange chromatography, or sorry, affinity chromatography, okay. Similarly, we are using oligo DT cellulose chromatography. I hope you remember about this. That will help you to separate mRNA from a complex mixture of the RNA, right? Okay. Along with you can use glutathione to separate GST tagged recombinant protein, okay. You can use also heparin, that will help you to separate plasma coagulation proteins, nucleic acid lipase. That's how various kind of affinity medias are available that help you to separate on the basis of their affinity, right? That's why the term affinity chromatography is used. So now let's talk about the advantages. So I have already mentioned that advantages here is high separation, extremely sensitive separation, okay. And the biological active site that we are want to use can be easily determined, okay. So we don't need to bother about too much, right? But it has some unfortunate disadvantages too. So what are the disadvantages? Obviously the ligand we are using will not be cost effective, right? So it is expensive and there is a chance of leakage of the ligand, and also there is a chance of degradation of the solid support. That are the various kind of disadvantages, and this kind of technique is used for vaccines preparation extensively. I have told you a example also that we are using this type of chromatographic technique in the level of in pharmaceutical techniques where we are producing GCSF, right? The example is NINTA chromatography. Okay, so along with in genetic engineering, in molecular biology technique, in another important pharmaceutical techniques, we are using this affinity chromatography. Okay, so today we have learned about affinity chromatography, that is nothing but a interaction of protein and ligand that we are using. So it help you to separate at high sensitivity, okay, but it have disadvantages, that is the ligand can be leaked and it is also very costly. Before that we have talked about ion exchange chromatography. So here we have discussed about cation exchangers and anion exchangers. Okay, so ion exchange chromatography's advantage is it help you to separate protein on the basis of their charge, okay, and the life of the resin is long and it is cheaper and it is dealt with only environmental friendly compounds. And the disadvantages are already the nature and interaction of the ion exchange resin and the nature of the exchanging ions, that are the disadvantages. And here we have revised about the column chromatographic technique and size exclusion chromatographic technique. So remember that size exclusion chromatography have a poor resolution, but column chromatography provides good resolution. In size exclusion chromatography, smaller molecule will be entrapped in the porous polymer beads, but larger molecule will be percolates through the solid matrix, okay, and it will separate faster than the smaller one. And the last one, that is our column chromatography. We have discussed that column chromatography helps you to purify and determine the number of components in a mixture, but column chromatography's disadvantage is the chance of air bubble entrapment, and also there is it takes too much time for separation, okay, and also column preparation also require technical skills, okay. And at very first we have discussed about these two things, that paper chromatography and thin layer chromatography, the very basic type of the chromatographic technique. So we have discussed up to that part 6 type of chromatography: paper, thin, then our column, then our size exclusion, and today's one ion exchange chromatography and affinity chromatography. So we have 2 last techniques, that is HPLC and gas chromatography, which I will try to upload within this week. As a result, our chromatographic technique will be completed again, okay. So I hope you are enjoying these revision classes. So thank you for listening to this class, and bye bye, take care.